In today's lecture, we're gonna see how, like, by activating the T cell, what is the result. So first of all, when the T cell will activate, T cell will activate when T cells will be activated. After then, the B cell will be activated. Okay, T cell, then B cell. So let's see how the T cell is being activated. We have done with the types of the T cells. Now we're gonna see how the T cells are being activated. So for example, this is the macrophage. Okay, this is the macrophage. Okay, and we know that macrophage, let me write, macrophage is a APSCS, means it is a antigen representing cell, means it represents it releases both the MHCs, MHC1 and MHC2. Like it has the both have both antigen representing, like it is known as the antigen representing cells. It can be represent antigen either by the MHC1 or either by the MHC2. So we know that MHC1 and MHC2 represent the antigen. So at this part either MHC1 can attach, or at this part either MHC2 can attach at one time, because it is the antigen representing cell and it is the ability of the macrophage. Okay, so by then, as we know that MHC1 and MHC2, what's the purpose of these are the special type of protein that represent the antigen, and then the T cell will read the representation and identify which type of pathogen it is or which type of antigen it is. So this is the work of the MHC1 and MHC2. Okay, so after then, when the MHC1 and MHC2 will be attaching to these macrophages, so what macrophage will do? It will release a chemical, and that chemical is known as interleukin 1. Okay, after releasing of the interleukin 1, the interleukin 1's purpose is to stimulate the T helper cell. After then, when it will be activated, the interleukin 1 will be released. So what's the purpose of it? It will release, or it will stimulate the T helper cells. Okay, it will release the T helper cell. So now this macrophage cell will be known as self non-self complex. Let me write here, why we are calling it self non-complex cell, because we are calling it because when the MHC2 will represent like this, when the MHC2 will represent the antigen, so that cell, means this cell will be called as self non-self complex. When the MHC2, listen carefully, when the MHC2 will represent the antigen, so then that cell will be known as, this macrophage will be known as self non-self complex. Okay, so now moving toward our remaining point, then the T helper cells will be activated, and what these T helper cell will do, these T helper cell will attach. For example, this is the macrophage and here is like the MHC2. So what T helper cell will do, T helper cell will attach to this antigen. As we know that MHC2 contains the antigen, let me make the antigen. Okay, this is the antigen. So T helper cell will attach to the antigen and it will read the antigen. What it will do? It will read the antigen. T helper cell will attach to the MHC2 when the interleukin 1 is being released. Okay, then it will attach the antigen and read the antigen. Okay, after then when it will read the antigen, so after that sometime the interleukin 2 will be releasing. What? Interleukin 2 will be releasing. Okay, so when the interleukin 2 will be releasing, so now which type of the T cells is going to activate? Now our cytotoxic T cell will be activated. Okay, cytotoxic T cell will activate, and plus who will activate? And B lymphocyte will be also activated. As I told you that first the T cells will be activated, then B cells will be activated. So now here, as the T cells are being activated, means T helper cells and cytotoxic cell are now the type of the T cells, so they are activated. Now the B lymphocytes, mean B cells, will be activated. Okay, then as we know that B cell will either make the antibodies, like it is divided into plasma cell and the memory cells. Memory cell will keep the record of that antigen, and plasma cell will do what? It will it will make the antibodies. Production of the antibodies. Okay, so now at this place, like when the cytotoxic T cells is activated, so what it will do? As I told you that macrophage. So this, let me draw that macrophage that we have drawn upstairs. So this is the macrophage cell, and that contains the antigen. For example, this is the antigen. Okay, this is the antigen. So either the cytotoxic T cell will release the toxic substance, it's the function of it, and or it will do the holes in it. What it will do? It will do the holes in it, like it will do the perforance in it. It will do the what? It will do the perforance in it. It will do the perforance like this. It will do the perforance. Okay, it will do the perforance in it. It will do the perforance, so that this antigen should come out. This antigen should come out by these perforance. This antigen should come out, and this is known as the this process is known as perforation. Okay, or sometime it releases, like it also increases, like releases the water and the salt. It sometimes this cells will like it's like the ability of cytotoxic T cell that it will also release what? By these perforin, it will also release the salt and the water, and due to this the atmospheric pressure will be high. Okay, but another condition is that here the here a enzyme is released, and that enzyme is known as granzyme. Let me write here. We'll discuss the purpose of it as well. Here the granzyme enzyme is released. What's the purpose of it? So granzyme enzyme will do what? It will do the apoptosis, means it will blast the cell. Apoptosis is a process in which the cell is being blasted because the enzyme is released, so it will activate, it will like catalyzes the process, and the apoptosis will occurs. And apoptosis, as we know, that the cell will be blast. Okay, when the cell will be blast, so the antigen will be also die. So this is how the T cells is being activated.